The effects of various metals on Alzheimer's disease in C. elegans. Alzheimer's disease is the most common form of dementia. Over 5 million adults in the United States are living with the disease. There is no cure for the disease, which worsens as it progresses and eventually leads to death. It is most often diagnosed in people over 65 years of age. Each year, over 83,000 people die from this disease, making Alzheimer's the sixth leading cause of death in the U.S. Alzheimer's disease is defined as progressive cognitive deterioration exhibited by memory loss. It is characterized by loss of neurons and synapses in the cerebral cortex and certain subcortical regions. This loss results in gross atrophy of the affected region, including degeneration in the temporal lobe, parietal lobe, and parts of the frontal cortex. In the early stages, the most common symptom is short-term memory loss. In the terminal stages of this disease, the patient will lose the ability to communicate, move, and at times is barely conscious. Neurodegenerative diseases such as Alzheimer's are caused by nerve cell damage and abnormalities in the brain by deterioration. This can be attributed to plaque buildup, which is caused by the protein amyloid beta. Amyloid beta peptides result from the amyloid precursor protein, or APP, which is being cut by certain enzymes to yield A-beta. A-beta molecules can aggregate to form flexible soluble oligomers, which may exist in several forms. It is now believed that certain misfolded oligomers can induce other A-beta molecules to also take the misfolded oligomer form, leading to a chain reaction. The misfolded oligomers and the resulting amyloid plaques are toxic to nerve cells. C. elegans are useful model organisms that can be used for the study of Alzheimer's disease. The transgenic C. elegans model has been used to study Alzheimer's due to its short lifespan, ability to develop muscle-associated deposits reactive to amyloid-specific dyes, and the progressive paralysis phenotype. Specific strains of C. elegans can exhibit amyloid beta proteins. The strain CL2120 exhibits amyloid beta proteins with GFP and can be affected by high temperatures to induce higher amyloid beta plaque amounts. Amyloid beta plaque in C. elegans can be studied using fluorescence and locomotion. The transgenic C. elegans model is a suitable model to relate amyloid beta expression with its toxicity. Copper, too, can be found in small amounts in the brain. In a study done by Hain, he used a 50 nanomolar copper-2 solution and found that it helped reduce amyloid beta plaque by binding to them. He concluded that copper-2 can help reduce amyloid beta plaque in cells and that compounds similar to copper-2 sulfates can also help reduce plaque, such as zinc and magnesium. Amyloid beta and copper play important roles in the pathogenesis of Alzheimer's disease. However, the behavioral correlation and molecular mechanisms of A-beta and copper toxicity have been investigated less often. In a study, Luo investigated the interaction and toxicity of A-beta 142 and copper in the A-beta 142 transgenic C. elegans worm model CL2006. His data showed that the paralysis behavior of CL2006 worms significantly deteriorated after exposure to 0.1 millimolar copper ions. However, the paralysis behavior was dramatically diminished with exposure to 0.01 millimolar copper ions. The copper treatment also partially changed the homeostatic balance of zinc, manganese, and iron. Luo's data suggests that the accumulation of reactive oxygen species was responsible for the paralysis induced by A-beta and copper in CL2006. Disturbances in intracellular calcium signaling may represent one mechanism underlying Alzheimer's disease. The penicillin-1 gene, or PS1, associated with the majority of early-onset familial Alzheimer's cases, has been implicated in this signaling pathway. In a study, Lise Ring used the Xenopus oocyte expression system to investigate in greater detail the role of PS1 in intracellular calcium signaling pathways. To determine which elements of the signal transduction pathway are responsible for the potentiation, he used fluorescent calcium imaging to demonstrate that PS1 potentiates inositol 1,4,5-triphosphate, or IP3, mediated release of calcium from internal stores. Lise Ring showed that an Alzheimer's-linked mutation produces a potentiation in calcium signaling that is significantly greater than that observed for wild-type PS1, and that cannot be attributed to differences in protein expression levels. His findings support a role for PS1 in modulating IP3-mediated calcium liberation, and suggest that PS1 mutations which contribute to Alzheimer's neurodegeneration may involve disturbances of this function. The purpose of this experiment is to study the effects of copper, calcium, magnesium, and zinc on amyloid toxicity in C. elegans using locomotion and longevity assays. The alternate hypothesis states that the various metals will affect the expression of amyloid beta in C. elegans based on longevity and locomotion. The null hypothesis states that the various metals will have no effect on the expression of amyloid beta in C. elegans. There are various techniques and assays that can be used to measure the effects of amyloid beta, including neurodegeneration, locomotion, and longevity. In this study, strains N2 wild-type Bristol and CL2120 will be used. CL2120 expresses GFP, human A-beta peptide, and accumulates of beta-amyloid fibrils at all developmental stages. A-beta toxicity is enhanced at higher temperatures. The strains were synchronized using a 20% hypochlorite solution. The effects of copper chloride, calcium chloride, magnesium chloride, and zinc chloride will be studied. A 20 microgram per milliliter concentration will be used for all chemicals. Each chemical will be inoculated in the nematode growth agar and E. coli food source. Using a fluorescent microscope, neurodegeneration in C. elegans will be observed for amyloid beta aggregation using GFP protein fluorescence. Images will be analyzed in Adobe Photoshop or ImageJ. Locomotion will be observed by counting the number of thrashes of a C. elegans per minute, with a single thrash defined as a C-shaped head-to-tail movement. Longevity will be observed by counting the number of dead C. elegans over a 10-day period. All data will be analyzed using SPSS software in which descriptive means, standard deviation, and statistical significance with a p-value of less than 0.05 will be found. Most of the materials used in this study are easily available and can be purchased through Carolina Biological or Sigma Aldrich. C. elegans and E. coli OP50 can be purchased from the CGC.